Very good morning. So today we are going to talk about the GLP, the good laboratory practices. So each and every diagnostic laboratory or hospitals or diagnosis center should follow and maintain these following steps to produce a very good result, because the doctors will prescribe the medication and the doctors will start the treatment on based on this uh, results. So this is very, very important. So GLP, is the good laboratory practices, is a quality system covering the organizational process and the conditions under which non-clinical laboratory studies are planned, performed, monitored, and recorded, reported, and archived. So these are the 6 areas which will be covered by this GLP. Okay, so the GLP ensures the quality and integrity of safety test data submitted to the government for the insurance of research permits. Okay, so the GLP only ensure the data which are correct, which are monitored during the test. Okay. So the two things are there, like a GLP, we have another one, GMP. So the GMP is a good manufacturing practices. So the GMP, the good manufacturing practices, applies to the entire drug manufacturing process, while GLP, the good laboratory practices, applies only to the safety testing phase. Okay, so the GMP is a manufacturing of drug, the GLP, the safety testing phase. So both GMP and GLP are enforced by the Food and Drug Administration in the United States, FDA. So the definition of GLP terms, the testing facility or the test facility studies, the research laboratory conducting the non-clinical study. Non-clinical study, and the test article or test item, the product being studied or tested are the subject of the system, subject of the study. So which subject we are going to study? So that regarding the article should be test. So the test system, these are any animal or plant or microorganism to which either the test or control article is applied. So the article, the control system will be one. These are the system will be applied for test. Any biological, chemical, or physical system used in the study. The control article or reference item, a product that is not the test article or a feed or a water, applied to the system, test system, are used to provide the basic for comparison with the test article. So the facility, what are the facility are needed? What are the facility are needed for GLP? The testing facility should provide separation of activities to prevent interference and other disturbance which may compromise the study. There must be a separate area for the receipt and storage of the test and control article. The place should be separated. So you can see in the hospital or the non-clinical area, the research center, the laboratory, the reception will be there. There they receive the article, and store. And the mixture of the test and the control article with the carrier, the storage of the test and control article or mixture, the housing of the test system, and the final one, all the equipments used in the study should be periodically calibrated and maintained. The records of calibration and maintenance should be kept and made available to operator of the equipments. So the records of the calibration. So calibration is very, very important to record, maintain the records. The maintenance should be important. Okay. The characterization of the GLP. The personnel performing the study should know the following about each test and control article. Each and every test should be contain the following three things. One, identity, the sample name, quantity, date, whatever it may be. And purity, how this is crude or a purity or purified. Or a composition, what are the composition present in the test article or sample. And the stability, this is heat stability, thermal stability, or the water stability, the stability, because this item will going to perform the equipments. And a date of receipt. Date of receipt. Now, what date the material should be received, because sometimes the same material will be received by the laboratory. So in that case, just the day we can identify the confirm with the date of receipt and the expiry date and the storage instructions. Because the laboratory number of samples will come, so we should know where to store, because we can perform the analysis or test on the day of receive or the next day or another 2 days, another 1 week. But the storage day instruction and the quantity received and the quantity used. For example, if the customer sent 100 gram of sample, so when we receive, we should make sure the quantity received, and we may use or either 20 gram or 30 gram or 35 gram. So if we supposed to use the 40 gram, we have to give the result. Along with the result, we have to return the remaining quantity of sample we received, because we whatever we charge for doing that for done or performed test, but accept we should receive. Okay. And the study plan or protocol. So it's very, very important of study plan. The study plan or protocol is the master guidance document for the conduct of the study. Okay, the outline how the study should be performed and contain the general time schedule for the study and its various stages. Also include the method and the material used in the study, which also include the protocol. Now, what are the method used for this particular test performed? So along with the result, we should mention in the report sheet this method we used for this testing sample and instrument. And the protocol must go through approval and revise and the discussion before the study begins. So before going to start the test, before going to perform the test, the protocol must be through approval. Okay, the test which will be going to perform in this technique, in this method. And this process starts with the study director preparing the protocol and discussion its content with the personal or other study staff. After discussion, the study director must then approve the protocol by affixing their date and signature. So once the discussed, the material should be countersigned by the director with a date, on which date the protocol or method or procedure is discussed and finalized for the test to be done or performed. And once the protocol has been approved by the study director, it must be reviewed by the QAU, the Quality Assurance Unit, who will assess its compliance with good laboratory practice. And at this point, the process personnel should be instructed on the duties assigned to them in the protocol, as well as received their own copies of the protocol. Okay. So storage record. So the storage throughout the course of the study, the study director will be responsible for the ensuring that the all data pertaining the study is captured, included, is record that the safety store. And these records are documents such as protocol, the final report, and the standard operating procedure will then be achieved at the end of the study. Only personnel authorized by the testing facility manager can access archived records. Additionally, every instance of records being accessed, removed from the retained to the archived must be live. And the example for GLP, the good laboratory practices. The below are general good laboratory practices. So wearing personal protective equipment, PPE, at all the times. So the PPE we can remember during the COVID, the public office and clinical workers on the nurse doctors, or we have some green color from head to foot. It's called PPE, the personal protective equipments. It's not only that time, it's very useful for all the times. And communicate with other members of the research lab, because not only one person will work in the laboratory, each and everyone should know the what is going to do. Participate in refresher training and safety exercises. So every person, so this is trial, this is practical, not only theoretical, they should participate in the refresher training, because the training only fulfill the knowledge of the methods and protocol, because it's a practical. We are going to do this in the practical. Now be aware of what you are doing at all the times, because it's whether the clinical or non-clinical or a drug or a food. So when we are going to perform the composition of the sample or quality of the sample, you should be aware of what you are going to do. Pay attention to unfamiliar smells and substances. Okay, so we should be very clear. We should pay 100% attention of what are substances and smells rise during our once they go over. Use the right lab equipments for the task or activity. It's very, very important. You know the procedure, you know the method, you know how to do, you have uh, chemicals and all, but the equipment should be perfect. It should be in condition. Then only you'll get the exact results. The regularly clean and calibrate and maintain the equipment. So the calibration is very, very important. And the cleaning is very, very important. So before and after doing uh, the test and the maintenance too. Okay, so these are the things, and the last, the 10 principles should be followed in GLP, the good laboratory practices. The test facility organization and personnel, and a quality assurance program, facilities, the parameters and materials, reagents, test systems, test and reference items, standard operating procedure, SOP, and performance of the study, reporting the study results, storage and retention records the material. These are the things should be followed in GLP, good laboratory practices. Whatever the GMP, the good manufacturing process will be or has been good during the perform the test. We should follow each and everything in correct pay attention. Thank you.